Okay, so on the screen, that's what we are going to do today. Remember, last week lecture, we didn't finish the last dimensionality reduction technique. It's called FIT. It's a new program recently published. It claims you can keep the original global structure between your data points. This claim is not true for t-SNE or UMAP. t-SNE, UMAP are stochastic based. It only take care of the distance between data points, whether they're close to each other or not. If they're close to each other to a certain threshold, you're more likely to be the linked neighborhood members. Otherwise, you just randomly put anywhere. So when you embedding your high dimensional data points using t-SNE or UMAP, you lost the global structure. What that means, if you have two data points are far away enough to each other, you don't consider it as members, then you can put them anywhere in your low dimensional space, 2D or 3D. That's why if you pay attention to some Twitter discussion recently, and people in population genetics using genetic diversity information from a human DNA sequence to put human individuals using UMAP or t-SNE to map the human relationship. And that was wrong, because if you have two individuals that differ enough, then you just randomly put them in the low dimensional space. And the t-SNE map and the UMAP will not reflect the real relationship between those individuals. So this paper published in Nature, I believe, has been criticized by machine learning field. And population genetics probably don't know the algorithm, the underlying algorithm. So they just use that to visualize the relationship between African, European, Asian, but that's the distance between those make no sense at all. After that, we will look at the k-means and dbScan spectral clusters, all built-in function in machine learning and statistic toolbox released by MATLAB, the company MathWorks itself. We can use their documentation, provide examples to look at those algorithms, and then we probably will use real single-cell data to repeat what we have, so to make connections between those very general algorithm of the clustering and see how you can apply to your single-cell data, okay? And then we will play with the SC3, and we have this function built in the toolbox. So through the interface, you will get a feeling of how long it will take compared to the k-means. Just give you some ideas of why it's justified or not to use that, okay? I believe Luis will have some demonstration of his optimization procedures, and I don't know how long it will take, Luis?
Very short, maybe 10 minutes.
10 minutes, great, okay. And then I have it's a guest lecture and from Michael Salins, and he's a postdoc in nutrition department. And we will not be here. I played the Zoom video, and he talked about the annotation of the cell type to give you the impression how exactly this workflow should work, okay? We have a one-click solution, and you get everything from the top to the end. But Michael will show you a real-world application. He uses his understanding of his data points, his cell type, and using so-called manual annotation. And I believe this procedure is very insightful, and we should learn the process and see how he can combine his prior knowledge about the cell type he knows from the prior literatures to identify those markers and to manually annotate each cell groups. This talk will take 20 minutes about, so that's our agenda for today. Is that okay? Okay, so this machine learning application, which is the method behind the FATE, the program. It's called manifold learning, and manifold is like a high-dimensional structure of mathematical objective. This object have a feature which these data points from this one to the other one, if you cannot jump, you have to go through the whole structure to reach that location. If you can see my point here, and if you measure the distance between this point in the center here to this point, it looks not too far away, right, from the Euclidean distance. But if you consider this manifold structure, you have to move small steps and cross all over this Swiss roll shape until you can reach this point. So that's why you cannot trust the directly calculate the Euclidean distance in high-dimensional structure. If you believe your data was somehow distorted in this more complex structure, you have to follow small steps and travel all the way along this framework, okay, this manifold to reach another data point. So if this linear relationship, the Euclidean distance, does not hold for this structure, so you have to consider different ways to understand the relationship between your data points. So that's a manifold learning try to do. And this method is start from look at the pairwise distance first, okay? So this is like a Euclidean distance we defined between each data point, so the pairwise. So once you have that, then you start to do a cutoff. If you have a distance very small, then you believe you can have one step or two steps to reach from one data point to another. But once you away from this threshold, and you don't consider you can directly jump from one to another. So using this so-called filter, the kernel function, and to filter out those long distance links between data. So after you trim off those long distance link, you leave up with this so-called local relationship, preserved connections, okay? So you learn from this structure to understand the relationship between data points, although the relationships are very complex. So this is the idea. So this is a paper published in several years ago, and becomes one of the popular application dimensionality reduction algorithm in single-cell data application analysis applications. So we have options. You have PCA, the very fundamental, hundreds of years old, and you have a t-SNE UMAP, which is relatively new and about 10 years ago, and very popular. And then this two, like 5 years ago, and you have three different major different types of these algorithms you can choose from. In the core of this method, it is MDS. Okay, I hope you still remember what MDS is. MDS is a multi-dimensional scaling. Remember, we have this map between different cities. Okay, you have from one city to another by train, how many kilometers, city 2 to 3. So you have this distance matrix. So dimensional scaling is dimensionality scaling is to build from this distance matrix to a regenerated map, basically, for the cities. So the last step of the FATE algorithm is MDS. But before that, we need to have a distance matrix between cells. And then you have distance matrix, and you use MDS to map those cells into the map. Okay? But before you cannot do this distance matrix directly because the relationship not reflect the real distance between cells. Some cell although looks very similar or close to each other, but if you follow the cell's development trajectory, you have to go from different states to different states, a lot of intermediate states. So this method starts from a distance matrix. So you have data and you compute the distance between cells, and then using this filter, okay, to transform distance into some terminology called affinity. So how closer each of these data points are. So through this transformation and use some diffusion probability method, but essentially the transformation or procedures to change the distance measures. Okay. After that, the last step is to use MDS to generate the map. So this is a very typical machine learning algorithm using a lot of this distance, kernel, affinity, diffusion, MDS, but you have to understand the core is to MDS itself, MDS. But before MDS has some distance transformation, that's all. So MDS allow you to keep the global structure, so the cell in this corner here will be very different from this corner here. So this map will be claimed that keep the global structure of the cell relationship. Okay, it's different from the t-SNE or UMAP. Okay, so I have my toolbox. So I want to show the t-SNE embedding, what's different from UMAP. Yeah, I made some modification for the interface, but essentially they are same. So you have import. I will use example data again. This time I use raw data without any processing. So no structure yet. And then you select this button here to select the different embedding algorithm. Okay, that's what you'll see. Okay, t-SNE, UMAP, and the FIT. All right. This is a relatively new, but it is slow, and that's why there's a turtle here. The publication you can read. This method basically try t-SNE, UMAP, and the FIT all together using different parameters, then take a consensus. Okay, that's why it's slow. It has to do quite a lot and then build another visualization. So now I will use FIT instead of other two. So you will see the result looks different. I hope no bug. Yeah, you can choose FIT. Yeah. So if you look at the behind this, the command one, the window, and those steps, MDS is here, diffusion steps, so everything is there. Stopped. Yes, it's still working on my computer. So later we will talk about the so-called pseudo-time analysis. So this is based on the projection of the cells. So if you have your cell after dimensionality reduction, you have a cells arranged like this. And in this case, you can build a hypothetical pathway. So you see this as my start point. I pick one of the cell from this boundary of this cloud, and then build a trajectory and go through most of the cells, and then I think this is the end. In this way, I can build a pseudo-time curve. So this curve can be regarding as a, say, if you believe this starting point like time 1, time 0, and then you move steps and to time n. Okay, you create a time points for each of the cell along this trajectory. I encourage you, you can do this after you finish fate embedding. Okay, you can find this trajectory here and maybe extract the cells to do this. But I don't suggest you to do this using embedding generated from a t-SNE or UMAP. Okay, so if you have X matrix and use t-SNE or UMAP, you create the embedding. Don't do pseudo time with that. Fate is okay. The reason is the fate keep the global structure for these cells. Okay? This is an algorithm guaranteed or built in there. t-SNE UMAP definitely no. Okay? Because once you move away from each other, there's no relationship between them.
How can you decide which one you choose? You just try 2, 3 different ways, and
there are many different ways, and you have used your own knowledge at the end. See, I want to use, I want to study one of this trajectory or this, so you can extract cells.
So embedding basically means clustering.
Yeah, embedding is before clustering. Yeah, so we haven't done any clustering yet. But this is a little bit jump, but I just gave you some idea that why fate is valuable, and okay, the developer making this really fast as well. So it's okay. Any questions? Any questions? Okay. So you can see a lot of the trajectories using fate. Okay. But fate is not very good for clustering, because clustering you want to have one class and another group, right? And if you apply k-means to this, you know, as we said in the lecture, k-means are not good to deal with this kind of structure. It's good for like one class. Okay, so in this case, I will change. I can use UMAP for demonstration, right? Yeah. Oh, you can, because this is a example data have already run through this, so you can just select existing ones and don't have to recompute. Yeah. You can recompute, then you get something like this, okay? So this is a beautiful 3D structure, and we can easily do this k-means algorithm with this. So it's a tree here, okay? So there are two buttons. One is so-called clustering using cell embedding. Basically, it's using the 3D data points, and forget about all the expression because we already embed or reduce dimension to 3. So the algorithm will only consider 3D position relationship between those points. And the button next to it is to clustering using original X, the expression matrix. So that will be the high dimensional, not only 3D. So this method, if you click this, this will be the only option I provided as C3 because I didn't find any other algorithms better than SC3. You have a lot. I mean, different publications you can make. Say I claim, but none of them good enough. So I only keep which reasonable. So we can jump. Let's cancel this first because according to our agenda, we will try k-means first. Okay. So that's what so we have fit and UMAP, two embeddings available for us. On this graph here is UMAP 3D embedding, so we can select this. And then we select k-means algorithm, and this is automatically generated. The numbers of k, remember you have to provide this k, and in order to let k-means to found how many clusters in there.
So if you use T-SNE, you cannot do clustering because there's no T-SNE is really.
Oh, you can, because if you run this using T-SNE, you will have that option because we haven't run that. If you decide to run T-SNE, then you have 3 options. You have a fit, UMAP, and T-SNE.
Oh.
Yeah. So if you select different k, still use.
Which was k-means?
Yeah, k-means if you use different k. So I will 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, maybe 10. You will get different picture, and so normally this is okay, and you can start to do the cell type annotation for each of these clusters. The one I just showed you, this very messy picture, this over cluster picture, because I want to make many small clusters and let algorithm to estimate or guess the cell types. And then if different clusters share the same cell type name, I will merge them later. You can also do this because in this case it's very clear this is one cell type here. So you don't have to over cluster to separate them, this one into 3 or 4 different subclusters and merge later. You can just directly say, okay, this is one cell type, and I will work on that.
So the number 10 means just compute 10 clusters.
10 cluster, yes. If you believe there are 11, then you can add 1. You can make changes here if you decide to say, okay, for example, I will use brush here to select these two groups. Okay, you see here there are this scarlet red and orange, these two clusters. I want to merge them into one. I can select and click merge. They should merge clusters and make 7, 9, these two, and merge into one. Okay. So you can make some small adjustment if you need. All right, any questions? Yeah, okay. Now I want to look at the k-means. If you look at the documentation, let's see which k-means. If you use which function, it will tell you where this function is, the file is located. Okay, remember the function file is end with .m, so extensions m files, and this function is located under the MATLAB itself, the toolbox and the statistics, okay, statistics toolbox within this. Yeah. We can open its documentation and look at the definition and how to use this method. Okay, so the basic use, you need at least two parameters. One is the data, okay? Data is big X. Remember this big X, we send it to big X to the k-means. It's not the original single cell expression matrix. What we send to it is embedded. So after this embedding, and we have, so this is we transpose it. Okay, so this will be cells by genes. Okay, so this original data matrix contains thousands of genes. But after we did this UMAP or t-SNE embedding, what we get, this X, like this small x, this data, that's our input for k-means. In this case, we still have the same number of cells, but how many variables in the column? Do you believe? Is this still the number of genes? No, because we have done this dimension reduction and it becomes 3D. Yeah, so this is S1, S2, S3. So if you use 3D embedding, each cell was defined by X, Y, Z. Okay, its location in your figure. So this is only 3D data. So that's what we send it to k-means function. So k-means take a small x, and the k is you define 10, for example. Okay, so that's our input, not the original. Can you put original in? You can, but maybe slower. Okay.
So that means you don't have to do embedding.
That's right. Yeah. 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 You should be able to directly give this big X to k. I believe so. Yeah. That's right. Yeah. I haven't tried how long it will take, but it shouldn't take too long. But this will be directly cluster your embedded cell data points rather than the original cell profile, and it will make more sense because you visually see, okay, this is a cluster, but if you use this, because there's a non-linearity within the cell, maybe you didn't cluster all the cells in that group in together. You may have cells in that particular cluster, looks same cluster, but the k-means give you different membership. Okay. So translate this into our function here. So let's see how we can do this. Okay. Assume we haven't done the clustering yet. Now you can export these variables. So now we need S. Okay, S is your embedding. It's basically is this. Okay, this is a small s. So if you want to do cluster based on this, you can export small s, and the small s, this variable contains the number of cells. We have 8,396 cells and 3 columns. Okay, these 3 columns define the position of each cell. So that's the variable we can give to k-means. Is that clear? Okay, rather than the original big X. The output is index. Give small here, okay? Then k, if I cluster them into 3D, okay? 3 clusters. No, not 3D, like 4 clusters. Then the output, the index will be either 3 or 2, 1 or 4. That's the indicator of the membership ID for each cell. Okay. So if you can get this. Is that clear? Okay? Yeah. So we run k-means rather than the original big X. We directly cluster its embedding based on the position of their embedding in the 3D space. And then we ask for, give me 4 categories, 4 groups. And this index variable will tell you, okay, the first cell is in cluster number 3, second cell is in cluster number 4, something like that. Okay, so that's what we need. Remember, you can plot this as 2, second column as 3. Okay, I generate a figure, a new figure, and using cluster 3 function to put my embedding into the 3D space. Okay, so that's what you'll see in this. Okay, you can still rotate this already. Yeah. Now I want to label different clusters because I have 4 clusters, 1, 2, 3, 4. I want to show them in different colors. I just need to put that index into this function. Because it's scatter 3 will take that. So I've put empty here and put the index here. This position is the size of your marker, okay? If you leave this blank, just like empty, I believe the program will use by default the 5 size of 5. So if you want to make this bigger, you can see, okay, 10 will have a very big bubble for each marker. But you can leave this like that. And the next will indicate different color, give different color indications. So if you run this, you should have these 4 clusters show in different colors. Okay. Yeah, let's consider where those information are from. So in our workspace, there are only 2 variables. The first one is S. This small s is how many cells and the position of each cell, X, Y, Z position in this figure, right? And the second variable is index. Basically, it tells you the membership ID for each cluster of each cell. So that's all the information we have. We put the position, first, second, third, the 3D position in this column to give this scatter 3 function, tell it where to plot those points. Then give this index to let it to decide whether you should use different colors to show the next point in the graph. Okay? If you run, for example, I rerun k-means. This time I use 10 different colors. And the index will change to indicate whether this cell belongs to group 1 or 2 or 3 or 4, 5, up to 10. You can regenerate this scatter plot also using color. In this case, you have 10 different colors in reality, okay? Okay, any questions with k-means? So if you go back to documentation of k-means, in the example MATLAB provided, is example fisheries, okay? So that's a 2D. Okay. I believe that's a different species. Then based on this 2D, the length of petal width, and then you can cluster different fish into 3 major subspecies, and then you can run those examples and cluster them into 3 groups. And this example is randomly generated 2 clusters, and then you can color them differently, and then also found the central noise. Okay. They use a function called the G cluster, similar to what I did, like 3D cluster with the index, the color. But they put the index here, okay? Okay, now we can come back here. Yeah, come back to here, and we will take a look of another DB scan. Okay, DB scan. Let's see which. Okay, in the same position, it's a function built in this statistics toolbox. Unlike k-means, DB scan requires 3 parameters. X is your input. You can give either this or small s. Then you need epsilon and the minimum points in the subclusters there. Okay, in here, instead of index, for example, instead of using k-means, I can try DB scan, say 5 different. It's not a k, so I need 2 numbers here. Epsilon, say 0.2, and 5 data points, something like that. I don't know how to predefine or determine those hyperparameters. Okay? So I just try this and see whether what it will give to me. Yeah, those 5 is the minimum numbers of this required. Yeah, so you change the parameter here, and like say 10. Okay, you require 10 members to be in the next radius as so-called. You can keep growing your clusters, and this will merge more cells. And I don't know how to change these parameters, but you get very different results like this. Okay. So DB scan, those numbers are not how many clusters you want to generate. I have no control. It's all based on data point itself. Okay.
So the 10 is neighbor points required. So you choose 10 points around this bigger sample. That's a small, right?
Yes. It's a different way to calculate or decide the number of clusters. It's not like deciding
It's
the
not
number of
number of clusters.
Mhm.
That means usually you don't use this small.
We don't know how to control those parameters, so we don't use DB scan at all. But it's one of the options if you have a density problem, because a lot of cells looks scattered around, but there are some cells very dense. Maybe some special cases we use DB scan.
So usually we use k-means.
We use k-means would be enough, yeah. Mm-hmm. But as a lecturer, I just show you their options. Yes.
So what is the numbers, like the 0.25 and the 10, what is that the parameters for?
This parameter tells you how you grows how you move your subcluster focus. So this algorithm will draw a lot of circles, and then within the circle, if you have, for example, you define 10, if there are 10 cells in there, it will consider, okay, this is subcluster. They start to move a little bit. If next point you have only 9 points, then it will stop here and introduce another new cluster, start to grow. If you make this number small, instead of 10, you say 5, and then this small bubble will keep going and until you stopped somewhere. There's no 5 points in that bubble. So this gives you some behavior control of this.
I
But
have a good combination.
Mhm.
You take epsilon 1.5 and the last parameter 10.
okay,
We come up with 10 clusters like this. I don't know how it works.
so 1.5.
Yeah.
Okay. So Kagada just tried these combinations. Then for these data points, looks like, okay, we get some good results. Yeah. Yeah, maybe in some case, you worth trying out some of the combinations of parameters. Okay, so let's try which spectral cluster. Okay, same toolbox. And look at its documentation. Mm-hmm. Yes.
So when we're trying to bring up the figure,
Mm-hmm.
put 5, then the IDX, what is 5?
Oh, this is the size of your marker, the circle in the plot. Yeah, if you want a big circle, you can pick a 10 or, yeah, control the size of this figure. Yeah, spectrum cluster algorithm allow us to have 2 parameters, input and k. And k, I believe, is the number of the clusters you can have. Okay. So same same as k-means, basically. Let's see, 6, let's try spectral. Okay, same. Okay, this is a result using spectral cluster and cluster cells into 6 groups. Maybe I change the number to 11. All right, this is just a very basic use if you decide to use command line to do this. Yeah, I believe all this algorithm you can apply to embedding. So this dimension reduced data set. So you can consider that as your input data set, even without, this one, because you already reduce the dimension to this, and you want to cluster cells. But you can still, if you decide to use original, but just remember the transposes make the cell by gene, and because those clusters, all these functions will take a matrix which rows are the cells and the columns are the variables to define those cells. Okay? Yeah, so for the input for those functions, either k-means or this or dbScan, they all take input and consider rows are the samples, are the cells. The columns are variables to define the properties of those cells. So if you put it wrong, if you transpose this S, and you will get an index only for 3 data points, because it will consider these 3 data. If you don't transpose this, put original X, you will cluster genes rather than the cells. In some study, you do want to cluster genes, see whether you can use the cells expression profiles to define the group of genes which express similar across those cell populations. Okay, so those are the 3 built-in functions in MATLAB built-in functions. Then here I want to close. Click this to close all other figures that only keep this original. Bless you. So now before the break, we can see, okay, I want to use cluster cells using this, okay? Use SC3, and remember SC3, the procedure. In here, I remember there's 1 page. Okay, so this is SC3. The input for this is this original, okay? It's gene by cell. So we don't need to embed the cell to 3D to give it. It will just use this directly to compute the distance between cells by itself internally. So that's why in this menu, the suggestion is to cluster cells using expression matrix X directly rather than the embedding coordinates of the cells. So here I say yes, this will take some time so we can have a break. And I just apply this, and then there's no, see how many options. How many we should have? 1, 2, 3, 4, 5, 6, 7. Let's say 12, okay? Well, something wrong, so we probably not be able to test this. Let me do a filtering. Mm-hmm. There's a bug in my code, so we cannot do it today. Okay, yeah. But anyway, we can take a break, right?
Are you showing the video that Michael recorded? Oh, okay. Because I messaged him and I was like, oh, are you turning? I hope not, because he asked me, but I never got back to the date and time. So I just wanted to tell him, yeah, it's a video. He was like freaking out.
Okay.
Yeah, all right. So Luis is going to show some demonstration on her computer on his computer to see the optimization procedures. Okay.
Yeah. Hello, everybody. Good morning. This will be very short. Just remember the pseudocode of the swarm intelligence algorithms. We have a search space and some particles trying to find a good solution that may be the best optimal solution, right? Just remember these meta-heuristics cannot guarantee that we are getting the best solutions, right? This is the trade-off. So you remember we have a function and we need to find the minimum value or the maximum. And here I have an implementation I did some years ago, actually maybe 8 years ago. So we have an implementation here, and okay, no, sorry. And we have some parameters. Actually, this program is running in C++. It's not the best framework to implement an algorithm, but we have as here the 2 dimensions. I will show you 2 dimensions. Some particles. These are the hyperparameters, and we can use, for example, 3 functions, 3 benchmark functions. For example, Rosenbrock. And this algorithm has some parameters, number of iterations, the particles to start. We can use more particles, but what is the difference? If we use more particles, I mean, some difference points in the function, we have the risk to get a sink into the search space. So if we use less particles, maybe we cannot converge. So this is the trade-off. So I will use 50 this topology of how the particles communicate each one or between them, global, local, focal. I will use global and some others coefficients. So I will graph GBES and PBES, and let's start the implementation. So you can see in the one step, let me slow down. In the very beginning, we have a random initialization. So the particles are trying to go to the 0 in that case. It's a minimization problem. So we can see, okay, maybe we need to, yes. We can see how the particles are moving this is a PSO actually. Is copying the behavior of the birds in the swarm. So here is the global best value, and it's not showing anything, but this is the average of the p-best. So at the end, I'm gonna increase the velocity. At the end, the particle is finding the 0, that is the minimum value, but is not getting sync because the particles are enlarging or are moving around the search space. So at the very end, okay, here we can see how the swarm is moving through the search space, and at the very end, so here we have 100 iterations. So let's increase the velocity so we can see how the particles are converging to the 0 point. So this is the beauty of this kind of algorithms, because find a value, but don't get or don't let the rest influence totally. So the particles can avoid to get sync. So we can see the GBest and the PBest, and okay, we can let the algorithms running, and maybe in the 2,000 or 3 or 5,000 iterations, it can get into the 0 value. That is in that case is the global optimum. So you can see is so yeah. So at the very end, so the algorithm is gonna find the optimal value. But in that case, this is the Rosenbrock, right? We can try a sphere. A sphere is simplest one. Is a simplest one function, and is the algorithm converge faster than in the Rosenbrock. So you can see, and goes to 0. So you can see the difference in the GBest, for example, it goes to 0 in the very fast in comparison with the previous one. So yes, so here we have the values in 0. So just remember the functions, the benchmark we are using. This is a sphere, is a very easy function to test, and the Rosenbrock is different, and this one is a raster engine. So what happens if we use raster engine, and we can set 3 dimensions, for example. Let's see how the behavior of the algorithms. Oops, let's decrease the. Oops, I need to stop and start again. So yes. So we can see this is the rastering function, but it's taking only. Let's see. Okay, we can. Well, this is not the best way to watch, but let me restart. Maybe there is some bug. And raster engine. Oops, sorry. Yeah, it's taking only a few particles, so at the very end, the particle is gonna converge in 1 point. So yeah, maybe the 3 dimensions is not working properly. So, but this is the general idea. Using particle swarm optimization is a swarm intelligence algorithm. Related with GA or genetic algorithms, I have an implementation using Python in Google Colab. I can share with you this file, this notebook. If you use Google Colab or Python, sorry, Google Colab is just an online platform to run Python code, and we can use for free. That is good. And we have we import the libraries, and we have the class. Actually, this example is in Spanish. I can't translate to English. Sorry, I was teaching in a Spanish class. So this is the individual or the gene, right? In the gene, we can map the values for our parameters. I explain here all the characteristics, and the initialization of the algorithm, actually I can run. The run is connecting. Yes, okay. I can run the individual. I have here all the actions of the interaction of the algorithm. For example, here I have the mutations operator, the selection, and some distributions. Okay, this is the population. So how many chromosomes or individuals inside the population? Let's see. Okay, I can go to the very end, just because the time. No. It's because I am explaining. It's very complex. I can reduce actually, and is easier. It's easier. So maybe it's from here. I create here the mutation, the mutation rate. Do you remember the entire chromosome, and the mutation rate is how percentage of the chromosome I'm going to transfer to the next one, or to use to create new individuals. So in that case, it's 50%. So maybe it's not the best example, because we can have a very high diversity. So I have all the values here to know what is happening for every generation, and here. So how I select the individuals, we can use tournament, rank, or a wheel. I forgot in English. It's a wheel. Is it randomly, for example? It's like using a wheel. So this is all the implementation. Yes, maybe I can need to reduce or simplify. So this is the function I am testing. This is my objective function, and I need to go to this point in order to maximize. Okay, this is a maximization problem, and at the very end, the particles need to come here, right? Using contours. Okay, is this function, but it's a different view. So the algorithms, this is implementation, and this is the best individual graph. So along the generations, the algorithms can go to the very top of the function. And yes, this is the video here. So we can see the genes moving across the function and quickly coming to the very top of the function, using this mutation or selection, mutation, and crossover operators. So this is the beauty of this kind of algorithms. Obviously, this is a very easy problem. So you can have a problem with is high dimensional problems, and you can mix, for example, with manifold learning, and yeah, you can. Okay. It's not moving the particles, right? No. But yes, as we see in the first example, so we can have the genes or the individuals moving through the search space in order to go to the very top of the function. Okay? Yeah. So maybe this example here is very complex. So I can reduce, simplify, and share with you, and you can use. You can open the notebook in Google Colab and run every cell or every part of fragment of code, and you're gonna see the program running. Okay, so this is the very short example I brought. So, okay,
Yeah.
Thank you.
Okay, cool. Maybe you can share the links to the teams.
Link of. Yeah, no, I will share the file. So you can open the file in Google Colab, and that's all. Thank you.
Thank you, Luis.
Thank you.
So now we are going to invite another guest speaker, from nutrition department through Zoom.
It's incomplete. It's still missing some data, but I did want to. Victoria's not here, but I did want to share it just so you guys can get an idea of what I'm doing while I'm sitting at my desk. So it's really the identification of all the different cell types in the mouse colonic tissue. So there are some data processing steps. The first one is filtering based on the QC plot, and I'll talk a little bit more about that and how I decide what to filter. Then it comes to the identification or the separation of colonocytes versus the non-colonocytes. These non-colonocytes are really just going to be the immune and the stromal cells that are also in these samples. And then once I can separate those, I can focus on the colonocytes, and I can then start to try to identify all the different subtypes of colonocytes in these crypts. And I'll talk again more about this in a bit. So this is the colon tissue. So we isolate this colon tissue. This is a human, but we get it from mice. And we have now started using the whole intact tissue. And what I mean by that is before we were just kind of taking off this top part, this epithelial layer, and then sequencing that. But we do know that down here is in the muscle layer is where all the blood flow is, all the immune cells are, and they are also responding to things like diet. So we're now interested in looking at the whole colon tissue. So we now have all tissue there. This is kind of what it looks like at a cross section. At the top here, you have the epithelial cells. These are the colonic crypts that are right here. And then down here, this is where you have your immune cells, your blood vessel cells, muscles, all different kinds of fibroblasts that give nutrients to the colonic crypt stem cells at the bottom. It's all there. So everything is important, and I think it's good that we have started doing this, but it does make the data a little bit more challenging. And as you can see within these crypts, there are a ton of cells really squeezed in here. And even in the center of the crypt, some of these circular things are part of goblet cells. So if we use a different staining, we can kind of see that the blue here are those goblet cells. And so we can capture all of this with our single cell data. So we have all of these different cell types here in the colon. This is just a simplified version of that. And this is what we've been using to do some RNA-seq. So the first thing, like I mentioned, is the QC filtering. This is really going to depend on whether you're using Flex or the 3' sequencing. The Flex is really clean data. You almost don't need to filter this data. If you do filter it, it would be just to kind of get rid of these cells that have a high mitochondrial percentage. But everything else here, to me, kind of looks fine. But if we come over here and we look at the 3' expression data, if we just look at the number of genes, this graph here, just from my experience, I've learned that down here in this section is really kind of junk cells. So we want to kind of filter this out to where it looks a little bit more like this over here. Even in the number of reads here, where there's this break right here, below that is kind of just these junk cells that we need to filter out before we begin processing the data. And that's how I filter my data. I have to look at the QC plots and kind of just go from there. Again, the Flex data that I'll be showing today is pretty clean, so I actually didn't perform any filtering on this data. This data comes from notobiotic mice, that is mice that don't have a microbiome. So this is pretty much just pure colon tissue, unaffected by the microbiome. This is what the data looks like when we load it into Dr. Sai's software, and then we can begin to embed it. I like using the UMAP because it just makes a lot more sense to me biologically compared to the other embedding methods that you can use. So the first thing when we have this plot, like I mentioned, is going to be trying to separate those colonocytes from the non-colonocytes. I use two major markers for this. I use EpCAM and EcadHerin. These are going to be expressed highly in epithelial cells. And this is what I use for me to separate the colonocytes, which are EpCAM positive, from the non-colonocyte cells over here. Of course, this isn't enough. I have to go in and really dig deep into what these cell types are. So just so that you guys know, about 60%, it's really about 50 to 66% are colonocytes, and the other percentage, 34%, is going to be these non-colonocytes. And again, this is going to vary. These are mice that don't have a microbiome. So in the normal wild type mice that we do see, this is going to change a little bit. There might be more immune cells because of the microbiome. I'll just really briefly talk about the non-colonocytes, but it won't be my main focus because these cells are actually pretty easy to identify. You can see that they make up just these little clusters here, and they separate really nicely, and we can identify them. And we get all different sorts of immune cells, B cells, macrophages. Sometimes I see dendritic cells, sometimes not. There's also blood vessel cells, so endothelial cells, smooth muscle cells, mural cells, and pericytes that are all connected to the blood vessels. Then there's all sorts of fibroblasts, some myofibroblasts, and then some other types of fibroblasts that, like I mentioned, are providing growth factors and nutrients to the colonic stem cells. And then we also see things like neurons and even adipocytes show up in this data. This is just a heat map for all of these cell types. So they do kind of break out really nicely. And I'm pretty confident in these cell types too, but I haven't put a lot of focus into this. I haven't really analyzed data coming from the non-colonocytes just yet, because my major focus has been on the colonocytes. So this is when I subsetted out the colonocytes, and then I begin to try to cluster these cells and see what are these cell types. So I use k-means clustering, and I kind of just play around with the numbers until I find one that really separates out everything kind of the way I like it, like visually, and then I can kind of go from there. So the first thing I do with the colonic cell types is going to be separating out the enteroendocrine cells and the tough cells, and that's just because they actually stick out from the data. So in this case here, this cluster 11, I know is going to be the enteroendocrine cells, and this cluster over here, number 3, is going to be the tough cells. And I know that by the marker genes that they're expressing as well, and we can actually calculate a score for these marker genes for the enteroendocrine cells. So you can see here they scored really highly compared to the rest of the clusters. That's cluster 11 up here, so I know those are enteroendocrine cells. And the tough cells, again, scored really highly and different from the rest of the cells. That's number 3 right here. So these guys are tough cells. That's easy. That's actually pretty easy and shouldn't be too much.
It's hard to me, but
like again, it's just because they actually pull away from the rest of the cells, because they are a differentiated cell type, but they are technically a specialized cell type as well.
Expression is so different. They just separate from the rest.
Exactly. The other cell types that I can try to pull out are goblet cells. Goblet cells are going to be this whole chunk right here. And I know that based on the MUC2 expression, so you can see that it expressed very highly in these clusters here. So I know those are goblet cells. I can also calculate a score for them, and they do separate out compared to the rest of the clusters down here. So I know, again, these are goblet cells right here. So I can actually subset out the goblet cells again, and then begin to focus on the specific goblet cell subtypes. I know nobody really does this, but I thought it was very interesting because once I saw this, I remembered a paper that I had read that actually mentioned that there's 2 lineages from the goblet cells. There's a canonical and a non-canonical lineage, and that's actually what I observed here, so that was pretty cool. Down here, what I'm showing is MKI67. This is a proliferation marker, so you can see that the clusters about right here are going to be the cells that are proliferating.
so that's interesting because they're supposed to be generally quiescent, right?
Yes and no.
So there are actually some cycling cells in there.
Yeah.
They're going to divide. Interesting.
There's also differentiation markers. So for goblet cells, a good differentiation marker is MXD1. And you can kind of see that, I didn't do a good job of lining these up right, but down here are going to be where those proliferating goblet cells are. And actually this little cluster right here that you see is secretory progenitor cells, or what I think are secretory progenitor cells. And then we have the non-canonical goblet cells here using these 2 markers that I grabbed from that paper. So this kind of branch right here is non-canonical goblet cells, and these over here are canonical. You can kind of see that this non-canonical branch is actually very close. That's this branch right here. It's very close to the other colonocytes right here. So it was very interesting to actually see that out on this 3D map. But based on those markers,
That's a biology.
yeah,
That's
For
right.
us, just two branches. We don't know.
yeah, I know, and I
You needed to read it.
I remember that paper. So for the secretory progenitor cells, they have different names and people call them different things. That's what makes this also very complicated. So I started calling them secretory progenitor cells. I believe previously we were calling them deep crypt secretory cells. So they do have this Reg4 marker. You can see that cluster number 12 is scoring really high for this Reg4 marker. That's this cluster right here. And you can also see that clusters 10 and 12 are also proliferating. This is a proliferation marker, so I know these are proliferating right here. DLL1 and DLL4 are expressed again in these secretory progenitors, and you can see that cluster number 12 is scoring high. It's actually the only one that's kind of scoring. So to me, these are secretory progenitor cells. This is what it looks like once you actually apply those cell types. So we have the secretory progenitor cells here. We have some proliferating goblet cells here, and then the rest, even though there's two lineages, I merged them just to simplify things. Those are the differentiated goblet cells. And actually, if we look at the differentiation potency, you can see that the secretory progenitor cells score really high in the proliferating goblet cells, and then the differentiated goblet cells at the bottom. So this looks good. That's what I expect from this data. Like I mentioned, we have these differentiated cells, but they're also very specialized cells. They have nice functions. And so you just saw that I was able to identify all these cell types here. The next thing would be to identify the stem cells and the absorptive colonocytes. So you can kind of see from this image to this image, I got rid of all those specialized cells. And now we're really left with the backbone of what I guess colonocytes do. So the colon is largely responsible for absorbing any sort of water that the small intestine didn't absorb. So that's what these cells, that's their function, and that's why they're called absorptive colonocytes. And so what I did was I subsetted out the stem cells and the absorptive colonocytes. They form kind of like this gradient, very similar, I guess, to this crypt right here. They form this gradient from stem cells and transient amplifying cells down here all the way up to differentiated cells over here. So I know that because of LGR5 expression is located down here at these clusters at the bottom. You can see that these are also the same cells that are proliferating based on proliferation markers. And then there's some differentiation markers that I use. I like to use KRT20 to identify the absorptive colonocytes. And aquaporin 8, this is, of course, a receptor that is responsible for pulling in water into the cells. And that is what I use to identify the late absorptive colonocytes. I don't believe I actually have an image of that, but this is what I'm currently working on right now. This is what I've had to do to be able to identify these cell clusters. My initial thinking when I first started all this single cell stuff was that, we'd be able to have our cell atlas with all our gene markers and cell types and just kind of, click and play and it'll identify stuff for us. I learned over a year or two that is not possible at the time, especially not for this particular cell type. It works actually pretty well for things that separate out really nicely, like blood cells, like peripheral mononuclear blood cells. Those do separate really nicely into T cells, B cells, NK cells, and I can actually identify them using a cell atlas. But for the colon, I just haven't been able to do that. So this is what I have to do. I just have to look at the expression across these figures here and then try to label these clusters here as a specific cell type. Again, this isn't finished just yet. I need to input a lot more details into this so that somebody can actually follow it. But for the most part, this is how we identify cell types in the colon.
Okay. So if you have any questions, I can help you to transfer your question to Michael, and maybe next time he has answer, I can let you know. But he didn't tend to give a guest lecture like this. He just report what he learned from the data itself. But it's really a good learning material for me, and to know how the biologists use their knowledge he familiar with all the markers he's supposed to see. And when he see these two branches, to me it's just two branches of data points, but he knows there's a canonical and non-canonical goblet cells. And so you do need to embed your biology knowledge in this analysis. And for those simple ones, you can just one click, T cell, B cell is okay. But once you get into the details of the subclusters, then you need those levels of the training to do this, okay? And also he mentioned in the middle of his talk some look at marker genes, so that individual markers. And he also mentioned some cell scores, okay? You remember, he said, okay, that scores is for calculate for each individual cells. Those scores were computed for combinations of multiple genes. So if you believe there are goblet cell, there are 5 markers, and how you can compute the score for regarding to these 5 marker genes to individual cell. That's what I'm going to demonstrate those and how can you compute, synthesize the score for each individual cells when you're giving a set of genes. Okay, those genes should be related or make sense to you. It's just not a random 20 genes. You can compute the score out of random 20 genes, but if you know what these 20 genes work together, do something, and you can compute that score for each individual cells, then you can differentiate cells into different subclusters regarding to the function of this gene set carried, okay? So if we come back to our example data, you can load any example data. Then there's a button, and it should be this. In the very center of the menu here, right next below the network, this. If you click this, and you will be asked whether you want to compute a score for those cells, or you want to compute after you want to compare 2 groups, okay? If your data have, say, wild type and knockout, you may want to compute the scores for all the cells that you want to compare between them. So in this case, I just say, okay, we just show the values, okay, for all the cells, not compare them. Then you have a selection of those, what kind of scores you want to compute. Okay, so this menu just let you to select different gene sets. Okay, some genes you want to study mitochondrial function, then you probably have 15 genes, okay, from the database, and you want to extract those names. So this menu allows you to select what kind of gene set you want to study to compute the score for. There are different options. I believe Michael select this cell type marker scores, and he want to study goblet cells. And he select this database, and then he selected this mouse data, and he select goblet cell from this database of marker genes. So I don't know how many marker genes associated with goblet cells, but yeah, we can find it out. But if you select this, the program will start to ask which algorithm you want to use to compute this score. You can select either of them. Okay. So the value is for every individual cell. Each cell now have one score. This score is the value is indicated by this color. And the yellow means that the score is high. So regarding to this goblet cell marker, I don't know, there are 11 markers, okay? This score is synthesized from 11 genes expression and look at those expression in each individual cells. And the values are higher in this cluster here. Okay, there's one cell that is really high. If everything is equal, then this cell with higher values are most likely to be the goblet cells. Okay? But you can run any cell type, but it has to make sense to the tissue. I don't know which tissue the cell we are using here, so maybe goblet cell is just a random score I computed, and the relative value different between cells, but not makes any sense here. But I just show you, okay, this is the way you can compute that. If you want to try other values, you can see, okay, I want to compute a signature score, and there are multiple options. Once you have select this, then you select whether it's a mouse data or not. Then you want to see, okay, you want to have a geo-terminology gene set, gene ontology. So you select M5, and from this long list, you say I want to look at regulation of cell growth. Okay, which cell, after computing, we have a highest cell growth related score out of those. All right, so if you learn how to click those, the next question for you is which one you want to select to understand your cell population. Then at that stage, I cannot help. I just gave you this, and you know. Just like Michael did, he know what cell type he want to look at, and then which marker he want to highlight. He can interpret those, either their differentiation or their proliferation, those signatures. Okay, so this is a very powerful tool, but you just need to know how to use it. All right. I don't have any other in our agenda, but I can leave this time for you for questions or any questions for Luis as well. Okay. Yeah, you can click give a robust test. If you run into any bugs, just let me know. Give me a screenshot. Normally, I can fix it in 1 or 2 minutes by looking at the code and change the places. So another technique is you can brush this cell population. See, I brush this green cell, and then ask for which cells are marker genes for it. Top 10 marker genes, and it should give me those genes that are highly expressed in the brushed cells. Yeah, so I brushed this particular cell type subpopulation. It's found, okay, this is PA, relatively highly expressed in the cell I brushed. But sometimes you don't find those markers, okay? So this is okay, maybe it's marker. All right. Okay, let me know if you have any other questions. And the next week we are going to talk about the clustering algorithms to classification. So this week is clustering, and then classification is you based on the training data, whether you will separate your data points into this group, that group. We will talk about neural networks. Kagata will give our discussion of neural networks. He's an expert on that topic. Okay. Yeah, Luis. Mm-hmm.